My name is Paul Proctor. I'm a master's student at the Scripps Institution of Oceanography at the University of California in San Diego. And I'd like to welcome you all to my talk entitled Phylogenetics of Swimming Acrocirridae, a family of polychaetes within the order Cirratuliformia. This video from Mbari shows a bomber worm, Swima bomboviridis, a gelatinous polychaete worm with unique bioluminescent bomb organs that the worm can release and could be a response to predators. They're about 2 to 9 centimeters long and are found between 1,500 and 4,000 meters. And this genus is named, noting their miraculous swimming behavior. The deep pelagic is underexplored and is an area of vast three-dimensional space. And thinking about a swimming 3-centimeter worm underscores the challenge of collecting what life may be lurking in the deep. In the past, deep sea collection was performed by trawling the bottom of the seafloor, including the first deep sea expedition by the HMS Challenger in the 1870s. And while there has been limited success, there are significant issues with this method. Pictured to the left is an acrocirrid identified through morphology that was collected from the deep and through key features was identified as a possible swimming species. This would be an important fact because it would represent the first pelagic member of Acrocirridae. But gelatinous organisms are often destroyed or badly damaged when collected, making these connections difficult to confirm. This challenge has been lessened by the advent of research ROVs and their increasing number entering the 21st century. That's provided new advantages to deep sea exploration, such as the ability to visualize what we are collecting specimens delicately and with intention, and the ability to do all of this at mid-ocean depths. Here's an example of another swimming acrocirrid, Teuthidodrilus being actively collected by an ROV through a high-flow suction sampler. This technology led to the discovery of the green bomber off Monterey, California in 2009 that I showed in the first video, and is the first proven pelagic member of Acrocirridae and a possible third origin of pelagicism within the Cirratuliformia, which includes Acrocirridae, its sister family Phylogenetic, and their neighbors Cirratulidae. And in 2011, another swimming genus, Teuthidodrilus, shown being collected earlier, was discovered in the Celebes Sea in the Philippines. It is distinguished from Swima by being slightly larger, darker, and instead of specialized bonds, it has long filamentous branchiae and large palps, which gives it a squid-like appearance, for which they get their name. Other pelagic discoveries were made at the same time, including Swima fulgida off Monterey, California, Swima tawitawiensis in the Celebes Sea, and the undescribed Tiburon bomber, horn bomber, and the Juanita worm off Oregon. Here is a map of localities that pelagic acrocirrids have been described from as of 2011. This brings us to the topic of my master's project involving 2 2020 expeditions led by the Schmidt Ocean Institute to survey the deep water canyons of Southwest and Northwest Australia. The ROV SuBastian collected 14 pelagic acrocirrid specimens with 5 morphospecies delineated using CO1 sequences and are depicted in this map. The Teuthidodrilus is shown twice for clarity. The first expedition to Southwest Australia took place from January to February and collected 2 unique specimens from 2 separate canyons, Perth and Hood Canyon. Sorry, backwards. The first specimen collected from Hood Canyon had a CO1 sequence that aligned closely to Swima, and it has specialized bomb organs shown by the arrow. The second specimen collected from Perth Canyon had a CO1 sequence that aligned to the Teuthidodrilus, and it has the long filamentous branchiae. The second expedition to Northwest Australia took place from February to April and collected 4 unique species from 2 separate canyons. In Clotes Canyon on February, 2 specimens were collected, and 1 listed as Swima species 3, circled in black, aligned to Swima by CO1, and the other circled in white, delineated with Swima tawitawiensis. And in March, another specimen, Swima tawitawiensis, was collected from Cape Range Canyon. Additionally, a second specimen of Teuthidodrilus was collected from Cape Range Canyon, and it shows the long filamentous branchiae and large palps as well. Lastly, 1 specimen was collected from Clotes Canyon and had a CO1 sequence that aligned to Swima, and it also has the specialized bomb organs shown by the arrow. An additional expedition that my master's work includes was conducted by the Nautilus Live Ocean Exploration Trust when they explored the Galapagos Rift in 2015 using the ROV Hercules. The team collected 2 specimens from that expedition that delineated with Teuthidodrilus same through CO1 and clearly had branchiae with a squid-like appearance. With these newly collected specimens, my next task was to construct an updated phylogeny, and here is the most recent phylogeny of Acrocirridae up until this point. I updated this by performing a maximum likelihood analysis with 5 genes, CO1, Site B, 16S rRNA, 18S rRNA, and 28S rRNA, with 10 starting trees and 100 bootstraps, and my preliminary results are as follows. First, the specimen from the 2015 Galapagos expedition is shown to be Teuthidodrilus same, confirming a range extension for this species. This is interesting development because it shows trans-Pacific connectivity for Teuthidodrilus same. I constructed haplotype networks to analyze the connectivity between these populations. There were 5 base changes in CO1 between the 2 populations with an uncorrected pairwise distance of 0.00792, revealing that there is some recent level of connectivity between these populations. Next, 2 specimens from Northwest Australia collected from Clotes Canyon and Cape Range Canyon confirmed a range extension for Swima tawitawiensis. This revealed clear connectivity between the Pacific and Indian Ocean through the canyons of Northwest Australia and the Celeb Sea, given the specimens from Clotes Canyon is 2 base pairs different from the specimen at its adjacent canyon and the specimen from the Celeb Sea. Next, the 2 specimens collected from Perth Canyon and Cape Range Canyon showed a high bootstrap support to be a new species of Teuthidodrilus. This reveals clear connectivity between the canyons of Northwest and Southwest Australia, as there is very low uncorrected pairwise distance, and the specimen from Southwest Australia has an identical sequence with 2 specimens from the Northwest Canyon. Our next result shows relatively high support for the specimen found at Hood Canyon of Southwest Australia to be a sister species to the undescribed horned bomber from Oregon. And another result shows high support for the specimen collected from Clotes Canyon, Northwest Australia in April to be a sister species to the undescribed Tiburon bomber from Oregon. Finally, the specimen from Clotes Canyon collected in February nestled in with all previously named Swima, but support is low to confidently put them sister to Swima bombaviridis, but I'm hoping that additional Site B data will help this support value and topology. Unfortunately, the Site B gene has proven difficult to sequence, but after some primer tweaks, we are hoping to have this data shortly. To conclude my results, the collection of 16 pelagic acrocirrid specimens, 14 from Australia and 2 from Galapagos, revealed 2 range extensions, 1 extension of Teuthidodrilus same across the Pacific, and 1 extension of Swima tawitawiensis from the Pacific to the Indian Ocean. My results also support the erection of 4 new species in the swimming clade of Acrocirridae. This includes 1 new species of Teuthidodrilus and 1 new species of Swima. These results also support extending the genus Swima to include 2 additional new species from the Australia expeditions that were sister to the undescribed species from Oregon. In conclusion, the majority of these pelagic worms have been found in the Pacific, but these findings have added localities from the Indian Ocean. Previously, 2 deep-sea genera described through morphology, Jovanelia and Hemetophorus, were described from the Arctic Ocean and Southern Ocean respectively, making those oceans interesting frontiers to survey with ROV technology. And the Atlantic remains surprisingly underrepresented given the amount of research that takes place. In conclusion, there are many more possibilities of where we could find these worms and bridge the gaps of evolution. For future work, I've gathered next-generation sequence data and used MitoFinder to extract mitochondrial DNA and construct whole mitochondrial genomes for 13 members of Acrocirridae and 2 pelagic flavulogerids. I've extracted the 15 mitochondrial genes and combined data from nuclear 18S and 28S rRNA to construct a more robust maximum likelihood phylogeny. Additionally, given that Acrocirridae has 1 swimming clade, I'm interested in studying the mitogenome for clues into the evolution of the shallow to deep transition of this group. A final step is to write species descriptions for the new species and potentially any taxonomic revisions to support the clade that my tree uncovers. With that, I would like to thank my advisor, my collaborators at the Western Australian Museum, the PhDs in my lab that taught me sequencing techniques, Dr. Marina Cowan and Avery Hiley, my collection manager at the benthic invertebrate collection, Dr. Charlotte Seed, the rest of the Rouse Lab, my guidance committee, Dr. Moira Decima and Dr. Ron Burton, and the captain, researchers, and crew at the Schmidt Ocean Institute and Nautilus Live Ocean Exploration Trust. And with that, I'll take any questions.